because Lynn basically made the whole introduction for what I want to talk about. Is it on? Yeah. Okay. So what I want to talk about is taking what Lynn was talking about to its logical conclusion. So basically, what can we do with life that haven't been done yet? And this is the great thing, is that she introduced the tree of life and this incredible diversity of life that we see all around here. But as most of you probably know, if you really look at life on the biochemical level, if you look at it from the point of view of the molecules that actually do life, we only have one form of life on Earth. And we're all coming from that last universal common ancestor, from that one particular form of life. And to make the matter even a little less interesting, the life that we know evolved in the gravity well of Earth, in this biological gravity well, but basically constrained by the environment, so constrained by what we have on Earth and what evolution has to work with. So this is the question that our field is asking. What else can be out there? What other ways can we do life? And I'm, like Lynn, I'm a carbon-based person, so I'm a carbon believer, so I will focus on biological polymers or things that you can do with carbon. And there's basically this incredible variety of things that we can do that life haven't done yet, and this is what we're exploring. So when you look at it from the perspective of a person that wants to experimentally approach the problem of the origin of life, we have a couple limitations. So we know that life started simple. It had to start simple. It had to start from simple molecules that somehow came together to create this complex life. But we only have one life form, so this is all we know how to work with. So experimentally, we've been trying to kind of approach this problem in two ways. One is people that are chemists like me take simple molecules, put them together until it starts resembling a simple cell. And then there are people that study modern cells, study complex cells, and try to go backwards, try to de-evolve it, try to look at what can we learn about the possible simplest life forms from taking apart the modern complex life. And there's obviously pluses and minuses to both of those approaches. If you work with simple molecules, you never reach the actual complexity of a truly living cell. If you work from a very complex modern living cell, you don't know when you got to this actual really simplest one. And so this is where the field of synthetic biology comes handy. We build cells, we build those so-called synthetic cells, which are cell-like objects, cell-like bioreactors, that resemble life and can help us model those very simplest life-like processes. And some of the things that we can do with synthetic cells are the things that are very relevant to this community. We can use synthetic cells as a model for the simplest life forms, and we can use them to investigate how life looked like on that early Earth. There are also a lot of practical applications for this field that mostly right now pays the bills. And so those two approaches to building synthetic cells, they're kind of a complementary, and they mirror what the people that work on the origin of life have been using. So there is this top-down approach where you take simple molecules, so things that could have been available prebiotically, and you put them together. So that's why it's top-down. You put them together into something that actually becomes very complex and lifelike. And then there are people that take very complex living cells and then strip them down, strip them of everything that's unnecessary until you start actually having something that could resemble the simplest living cell, the most primitive living cell. And when those two approaches actually meet, that is what we call a synthetic cell. When we actually put something together that's made in the lab, that's made experimentally, and is a living cell. So just to give you the punchline that haven't happened yet, and I'll show you in a second why. So what is a synthetic cell? It is something that is more than a sum of its parts, so it has emergent properties. It's not just a biochemical experiment, but you can still control it. This is the key. Normally when we work with biological experiments, we cannot fully control our system. We cannot control every single molecule that goes in there. With synthetic cells, we can. That's the biggest selling point of it. Everything that's controllable in a synthetic cell are those parameters that you want to be able to control if you want to build, for example, a titanium or a European organism that could evolve on Europa. Those things that are elemental to metabolism that we cannot control in living cells. So that's why I'm saying what I'm doing is taking Lynn's experiments to its logical conclusion, because I step out, I actually like the word chassis, I step out of the chassis, of the biological chassis, and I control things that cannot be controlled in the natural living cell. So for example, things like amino acids that are the basic building blocks of life. We can use unnatural amino acids. We can use X and A backbones. We can control size and shape of the cell beyond what normal cells allow you to do. So this is the system that I work with in my lab, and that's essentially a soap bubble that makes proteins. There is a liposomal bioreactor that contains the central dogma. So right now we're using mostly elements that came from natural biology, because that's what we have handy. We want to step out of that, we want to expand it towards those more unnatural building blocks. So the membrane of most synthetic cells is just a lipid bilayer membrane. It's most of the time a membrane that's made of phospholipids. So these are these guys, mostly diacyl chains with a functionalized head group. These are essentially the lipids you guys all have in your cells. These are the lipids that modern biology uses. Sometimes when we want to go towards more prebiotic, plausible experiments, cells that looked like cells on the early Earth, we go to the simple non-functionalized fatty acids. So this is more like the lipids that we could have available on the prebiotic Earth. So this is the compartment. Sometimes we give up on the membrane altogether and we use those so-called droplet microfluidic systems. This is a video from Theo, a postdoc in my lab, where he makes millions of those essentially identical synthetic cells. Here they make GFP in those microfluidic systems. But regardless of how you encapsulate it, the really interesting part is what goes inside. So this is the metabolism of a synthetic cell. And what we use is the so-called cell-free protein expression system. So right now it's mostly guts of bacteria. You can use guts of any other organism you want. People make cell-free systems from pretty much any domain of life. And if you tune in on Thursday, I believe, Vincent Moira will give his talk. He will tell you much more about cell-free systems and how those systems operate. We basically use them to make proteins. So we follow the central dogma of biology and we use them to make the cytoplasm of synthetic cells. So once you make that synthetic cell, you can do a lot of really cool things with it. So for this community, what's really important is this part of the graph. So we can use synthetic cells to understand how natural biology works. And we can use it to do things like study the origin of life. So we can use them as models of simplest cells. We can use them for long-term space exploration for things like astropharmacy applications. And we can also use them for a lot of kind of a practical, currently applicable purposes like medicine, like building tools for studying biology. For example, those of you who do any molecular biology, you probably heard of so-called Gibson ligation protocol. It's the most commonly used cloning technique these days. And that was developed by Dan Gibson when he was working at JCVI building the first mycoplasma-based synthetic cell. That shows you this cross-pollination between synthetic biology, people that make synthetic cells, and all the other areas of biology. So here's a quote from New York Times from October 9, 1903. New York Times is a very respected publication. They published this article where the author was arguing that it's absolutely waste of time and resources to try to build a flying machine that's heavier than air. The reason I'm showing it to you is probably a lot of you were listening to the last few minutes of my talk saying this is complete nonsense. You will never be able to build a synthetic cell that took hundreds of millions of years to do that on Earth. How can I hope to do it in a lab within a finite time? Well, people always think that. People think that the progress will never happen because it's too hard to imagine it. That article was published in October, and two months later, the Wright brothers flew their first Wright flyers. So that shows you that if people tell scientists that something is impossible, we are like the kids that Lynn mentioned. Reverse psychology really works well on us. Someone tells me it's impossible to build a synthetic cell, I tell you, okay, I will build it. So now I will show you, the next few slides are going to be the only actual work from currently happening in my lab. I'll show you where we are in this process of trying to build a synthetic cell. So what we did is we engineered a complete cell cycle of a synthetic cell, and that synthetic cell can undergo natural selection. And so this basically shows you what those dead as a doornail systems, this is not a living cell, this is a biochemical reaction system that's put together to look like a cell. What can it do? So what we have is we have a genome inside a phospholipid vesicle. So we have a membrane, inside that we put DNA genome, and that DNA genome can be replicated. Now that DNA genome can be expressed, this is the cell-free protein expression system that I mentioned. We make proteins on the template of DNA, so basically express DNA to proteins, just like in any almost central dogma. We have membrane proteins that go into the bilayer of the membrane. We have those membrane proteins then are used to secrete waste and to uptake nutrients, uptake small molecule nutrients. So that cell is a perfect heterotroph, it makes nothing, it has to get all of its nutrients from outside, and it has to release the waste to the environment. Then we have the feeding. So synthetic cells, like every other organism, get hungry and they eat. And what we did is we engineered a feeding mechanism for synthetic cells that genetically encoded. So basically those synthetic cells, as a result of a gene that's expressed inside a synthetic cell, they uptake feeder liposomes. We have those little liposomes that we feed to the synthetic cells as food. And the synthetic cells uptake those liposomes by a membrane fusion mechanism, and that fusion mechanism is encoded by a gene that's inside a synthetic cell. Now, if you have something genetically encoded, that means you can mutate the gene that's responsible for it. And this is our natural selection. Because the feeding is genetically encoded, if you use a gene that produces a better ability to uptake nutrients, then some synthetic cells that have that gene will grow bigger, and the ones that have the weaker feeding gene will remain slightly smaller. Then you divide it. This is the most artificial part of the system. They do not spontaneously divide. We have to induce division. But when they divide, they produce daughter vesicles. They somehow always call daughters. There are no son vesicles for some reason. And that I didn't make that up. That's how I was trained, that when you divide vesicles, they always produce daughter vesicles. So we produce a lot of daughter vesicles. The ones that ate a lot end up fatter right before division, and they produce more daughter vesicles. That's how your beneficial mutation spreads through the population. And then last but not least, there is a generation counter that we had to develop just to show that the synthetic cells that we're working with actually went through several cycles of that growth and division. So this really simple system that we managed to reproduce in the lab, we can show 5 generations of growth and division, and so on. We can show that there is this lineage of synthetic cells that can undergo 5 generations of complete cell cycle with replication of DNA, growth, division, feeding, waste removal. And we can show competition, where a gene that allows them to grow faster, allows them to produce more offspring, and that gene takes over the population after a few generations. So we can show this very primitive natural selection. Why is this not yet living? So I subscribe to the NASA definition of life, which is a self-sustaining chemical system that's capable of Darwinian evolution. And our system right now is not yet self-sustaining. This is the key. One can argue that no life is really self-sustaining. If one of you was put on the surface of a completely sterile planet right now, you would not remain alive for long. So does it mean you're not alive, you're not self-sustaining? You know, another definition is that life has to be able to self-reproduce. I cannot self-reproduce. I need my husband to reproduce. So I am not alive according to that definition. And so this is all kind of getting into this what is life really, and at which point we become alive. I am willing to argue that this system that we built undergoes complete cell cycle, undergoes a evolution, but is not yet alive. And that's something that I'm happy to discuss with, especially with physicists. For some reason, physicists told me that this system is already alive. Biologists think it's not. Funny distinction. But anyway, this is where the field is right now. We can build this, again, completely dead biochemical system that can do all those things that right now, we used to think only life can do. So what is it good for? From the point of view of looking at the emergence of life on Earth and looking at how life could start on Earth and how life could start elsewhere, this is the perfect experimental system that lets us basically do what Garrett in Lynn's lab is doing, except take it again to its logical conclusion, move away from that biological chassis and, for example, try to figure out how evolution of biotic compounds, so things that can emerge from carbon in water at certain temperature, how could that assemble itself into, for example, a life on Europa? So making a European organism, or European I think, making an organism like that in a test tube without the limitations of what is the current chassis of life on Earth. So forget bacteria, let's just make something that sort of looks like a cell. So we're still staying within the framework of what this guy saw under the microscope 300 years ago and say, oh, that looks like a monk's cell, let's call it a cell. We're still there, we still have a compartment that has biopolymers inside, but now we can basically do that evolution any way we want from scratch. And then another kind of an astrobiological application of it is that if you make a cell, you can then write down the exact composition of that cell. You cannot do that with a natural biological cell. We can write down the composition of our synthetic cell because we make it from scratch, from elemental components. If I write down the composition of something, then I can send it to a remote lab location without having to physically send you reagents. And that becomes handy when the remote lab location becomes somewhere where FedEx doesn't deliver. So this will allow, for example, doing support experiments for long-term space missions. This will allow using synthetic cell chassis to do biomanufacturing for long-term space missions. So some people here talked about going to retrieve an asteroid. One day we will actually want to go there in person. One day we will actually want to send people there. And once we do that, we need this biomanufacturing platform that will help them stay alive. And that's one of the kind of good applications of this kind of a crazy synthetic biology work, is that we can do something that's not possible with natural biology. So now I really want to quickly go through 4 different examples of what synthetic cells are already good for in this realm of practical applications that will one day become useful and relevant to all of you. So synthetic cells have already been shown to be able to shrink a tumor in a live mouse. This is work from Avi Schroeder at Technion. They've shown that a breast cancer tumor model in live mice can be shrunk by synthetic cells. So this is the new types of therapies that we can have. Synthetic cells have been shown to induce growth of vasculature. Another really practical application of this technology that one day might help with long-term space missions. Synthetic cells have been shown to be good platform for making vaccines that can be stored as in a freeze-dried, lyophilized state at room temperature for a long periods of time. They've also been shown to be able to make replication-competent bacteriophages in that system. So you basically have all those applications that are already possible with this very simple cell-like system. So as every new area of science, we're doing something that's kind of crazy. When you think about it, making life from scratch, whether for origin of life or looking at life under very extreme environments elsewhere in the solar system, that is the ultimate gain-of-function experiment. So how do we do it in a way that doesn't create another pandemic? That's obviously something that we all care about. And that's another part of my work that I'm really passionate about, and I just want to kind of give you a really quick overview of what our field is doing to not end up killing everyone. And so what we do is we follow biosafety, biosecurity practices as we develop this field. So basically, as I work on every new application of synthetic cells, I do dual use concern analysis, I do mouse analysis, we do consultations with people that work in biosafety, biosecurity. We do work with every stakeholder in this field, so not just the research community, but also people that pay for our work, people that do citizen science, people that actually use those technologies to do those experiments, fun biological experiments that they do to kind of show the premise of the field, and the general public. Outreach is a really important part of this work. We're building life from scratch. That sounds obviously fascinating if you're inclined to be excited about science. If you're inclined to think science is evil and it shouldn't be done, then that's something that we really have to work on our public message. We really have to show people that what we're doing is practical applications, not just for esoteric applications like origin of life and astrobiology, but also for practical biomedical applications. We need to talk to those people who actually might have concerns. We need to address those concerns preemptively. And so yesterday we were asked this wonderful question, and we were shown this example that if you don't ask for money, you're not going to get it. So we're asking, we as a community of people that engineer life from scratch, we want to ask for a giant investment from our governments, from big private foundations, and other fields have done it. We've seen all that nicely summarized yesterday. Scientific communities managed to come together to ask for billions of dollars. What is that we as a synthetic biology community, people that want to practically study the earliest evolution of life on Earth, that practically study the possibilities of evolution elsewhere, what is that we could ask for? I do not have an answer to that question. I always like to ask that question, and maybe someone, basically we want to narrow down this list of asks and priorities. And when I say we, what is we is the community, international community of people that think building synthetic cells is not only possible, but also necessary. It has to happen. And so as Lynn introduced our Build a Cell community, I just want to tell you there's literally dozens of us. There's people that work on engineering synthetic cells on most continents. There is this amazing community of people that believe that getting outside of this natural biology constraints of natural evolution is the way we have to go if we really want to use biology, not just to understand the origin of life on Earth, not just to find life elsewhere, but to survive as a civilization, to be able to actually do biomanufacturing, to replace oil with biomanufacturing. We have to make new organisms that will actually start doing making those chemicals that we're right now getting from oil. That's just kind of one of the examples of the importance of this work. Why making cells, synthetic cells, pushing the boundaries is not just important for what we're doing, which we all find fascinating from the foundational science point of view, but we also need to do it. And so this is our international community, Build a Cell. What we do is we do not define life. We do not say this is the only way to do synthetic cell. We want to be really inclusive. We want to coordinate research. We meet as a community twice a year on our workshops. We do outreach education. We try to work on science policy, so science policy actually keeps up with experiments. Right now we are funded by NSF, thank you NSF, we are Research Coordination Network, but we're also international despite being funded by NSF. We have members, Build a Cell has members from a lot of different countries. And if you want to learn more or if you have any interest in this community, please talk to me, or please talk to Lynn, or please talk to Vincent. They're both on the steering group members of Build a Cell, they're both here, so any one of us would be very happy to tell you, please come join us, we have cookies. So the community is really friendly. What we do is not just make life from scratch, but also try to find applications for it. And this is the last but not least thank you slide to everyone who's doing work in my lab. I've been a PI for almost 5 years now, and it's been an amazing wild ride to be able to start a lab and then say I want to make a cell from scratch, and someone actually gave me money for it. And then someone actually joined my lab and decided to do this work with me. So I'm immensely grateful to all those people that have been working with me, have been in my lab, and then all the collaborators that I don't have time to name here. So thank you, all of you, and I will take questions.
Yeah, of course. Hi, thank you for this very fascinating talk. So I'm an astronomer, so I might ask you the naive question. So this thing that you built, this synthetic cell, you said that it's not living, and if I understand correctly, you have to induce division, is that correct?
Yes, so that's why I think it's not living. Some people disagree with me on that point, but to me, to live, you have to at least decide you want to divide.
Okay, so basically you have it here, you give it food somehow.
Yes.
And then it lives its life, but it's not going to reproduce. Is that what happens?
Yes, it will grow, but it will not start making daughter cells, at least in the current form. People have done work on cytoskeleton engineering, and they can make synthetic cells to look like they're almost about to divide. They can engineer them to the point where they have those contraction rings. They look like budding yeast, but they do not finish division yet. To me, that's important. Some people think that cell cycle is already enough for life, but I'm a replication purist, I guess.
And by curiosity, how do you induce division, and do you take a knife and cut?
Yeah, I take a lot of little knives. It's a polycarbonate membrane that has pores in it. And when you extrude a liposome through that membrane, it divides. That's just one of the ways in which this can be done. You can also do it by mechanical agitation, and some people do that like that, but we do it by this extrusion process.
Thank you.
Thank you.
Hi. So the next naive question, bordering on unfair, is it has to do with information. So if the origin of life is the origin of Darwinian evolution, well, the origin of Darwinian evolution is the origin of information. Where does the information come from? And if it's the information that you provide, right, in the synthetic cell, then it is not a cell from scratch.
Yeah.
It's a Frankenstein.
It's a Franken cell, we call it like that.
So I'm on to something. So with that understanding, this is the unfair question. Where does that information come from?
Oh no, it's absolutely fair. Right now it's coming from literature.
No,
we don't create it.
No, I'm talking about in natural system. Where did that information come from to instruct the chemistry to begin the first step towards Darwinian evolution? There must be an information source in nature that is regularly spaced, probably in charge, probably in solid state. And I know that there are several of us here in the room. I'm looking also at Eric about this, who think about this. But I would like to add that to this discussion, and I would appreciate to hear your thoughts on that.
My thoughts are that I'm not actually an expert to answer that question. All the work that I'm doing, I'm basically a pipetting monkey. What we do is we put together things from components that we already know will work at this point. When we move beyond that, we will be putting together components and evolving them. So we'll basically be making libraries. I don't know where that came from at the very early stages. If you think about like the very origin of ribozyme evolution, for example, you can think about making giant pools of random RNA, and in that pool, at some point, there might be a self-replicating ribozyme. But where that information came from, I really don't know. I think that we need a talk where someone uses the word Shannon entropy every other word to explain, really. And I think you had a really good point that Eric and other people here would be much more competent to answer that question.
Okay, thanks again for that beautiful talk. I wonder, because you work with liposomes, let me introduce myself, Petr from Sofia University. So, what do you think of so-called GARD model? This is a model of, you probably know that, of evolving life only based on lipids.
Yeah. So, I did my PhD in split between two labs. One of my PhD supervisors was a peptide lipid guy, and the other one was RNA world guy. So I've heard both sides of that discussion before I was able to form my own opinions. I honestly, just looking at the modern life and trying to go back in evolution, I'm a big believer in RNA world and lipids that came relatively early. So to me, the individuality that's necessary for the origin of Darwinian evolution was conveyed by encapsulation. And I was very careful when I say that's my belief, because we obviously don't have, until we build a time machine, we will not be able to say for sure how that happened. So to me, the most plausible scenario of the earliest biological evolution on Earth is RNA encapsulated with small peptide helpers inside a liposome. I do see the value in people that draw those scenarios of membranes being the catalytic center, especially if you look at things like the electron transfer, for example, mechanism. That's one of the things that those people use as an example of that membranes came really early and drove the metabolism. To me, that's really attractive, but I don't see the way how from that you could make a step towards then encoding it. And if Darwinian evolution had to happen, like you said earlier, it has to be information, and that information has to be encoded in some sort of a genetic polymer. RNA is an accident. We could have any other polymer like that, but some sort of a biological polymer had to be there from the very early. So my personal opinion is that RNA world then got encapsulated, not the other way around. But we won't settle it. I mean, we can do experiments that prove both versions, but it's not going to show you what actually happened on the early Earth.
Can I ask you, have you ever watched Jurassic Park?
Yes,
you're doing dangerous work.
Life finds a way, I know. There can always be beans that the synthetic cells eat. So,
let me ask the actual question. That was low.
It's a very good point, though.
Yeah. What about reverse engineering and finding Luca?
So you're talking about things like going back phylogenetically, trying to do ancestral gene.
Yeah, recover last universal common ancestor.
So I think this is something that I know that Anton has a talk here at some point later in the week. I suspect he's going to really talk about that more. What we're doing, at least right now, is we're trying to get anything to work, which means we're trying to build a living cell from non-living components. We're not fancy enough to do ancestral gene reconstitution at this point yet. Where it does come into play is doing things like, for example, ribosome evolution, and you'll hear much more about that later in the week when Anton and Lauren talk. When we build the synthetic cell the way it is right now, it's too small to be able to allow enough robustness to use ancestral genes. Because most of the time when you do ancestral gene reconstitution, you end up with a gene that has much less activity than the modern counterpart. Our systems are not efficient enough to accept less than best possible enzyme for every transformation. And one example for it is the genome replication that we're using. Right now we're using a 529 DNA polymerase. It's a polymerase from a phage. It's a highly evolved polymerase. It would not work with any less evolved polymerase right now, but we're hoping that once we build this system up a little more, we will be able to build in that robustness, and then I'll be able to do ancestral gene reconstitution and try to do a primordial polymerase. We've looked at that, but right now it just would not work in this system.
I have a simple question. Is your cell in thermodynamic equilibrium?
What do you mean?
Is it in equilibrium?
Right now it is not in equilibrium, and that's because we on purpose push it out of that equilibrium. So we deliver energy, high energy molecules like ATP.
No, you feed it. That's a feeding, you mean?
Yes.
But
yeah.
before you feed, it stops and it's in equilibrium.
Yes.
Then you insert in artificially.
Yeah, exactly.
So the system in principle is near equilibrium all the time, and you move it out every time by the feeding.
Yes, I believe, with my limited understanding of physics, I believe that's how it is. It gets pushed out of equilibrium every time. And then the same for division. When they grow, they stop growing spontaneously at some point, and they just stay big, and they just float there around as perfectly happy, chubby guys. And I have to extrude them to make daughter vesicles, so that also could be understood as pushing it out of equilibrium.
Second, do you have DNA inside the
Yes,
cell?
we have DNA.
Do you have repair enzymes?
No, we do not have DNA repair, which is part of the reason why we've only gone through 5 cycles of growth and replication. We do accumulate mutations. It's not a high mutation rate, that polymerase is very, very high fidelity, but we do eventually see degradation of our genomes, and one of the next steps would be to try to do some sort of a proofreading and DNA repair.
Okay, thank you, Kate.
Thank you.
Clearly a ton of enthusiasm and to the audience.